Welcome to day 2 of the CAS-MS conference. My name is Darren. I'm co-chairing this year's conference together with Jason. We had a fantastic day 1 of the conference. Thank you all for the active participation, and let's keep the momentum going. It is my great pleasure to introduce today's keynote speaker, Dr. Rohin Mhatre from Biogen. As many of you already knew, Rohin has worked in the pharmaceutical company industry for the past 30 years. Through his 20+ years tenure at Biogen, he has held various roles. Presently, he is the Senior VP of Product and Technology Development. Prior to that, he was the VP of Global Regulatory Affairs CMC from 2015 to 2016. And from 2009 to 2015, he was the VP of Biologics Drug Substance and Drug Product Development, Analytical and Pilot Scale Manufacturing. Rohin graduated with a PhD in chemistry from Northeastern University, and also he served as an adjunct faculty there as well. Rohin has several publications in the area of protein purification and analytics, including co-edited book on quality by design for biologics. I also want to emphasize that Rohin is one of the original founders of CAS-MS conference 18 years ago. So welcome back, Rohin. Now I will turn the podium to him for his keynote presentation, Continuous Verification of the Next Generation Processes, Utilizing Enhanced Process Control. Please go ahead, Rohin.
Thank you, Darren. Really, hello to everyone. It truly is a pleasure to sort of see where this conference has come to. And I was just amazed because when we kind of started at this point, 18 years ago or so, this was just an idea. We thought, let's get together, chat a little bit about mass spec, see where that leads us. And I can't sort of be more appreciative of what number of people have done. Jason, who I worked on from very early on to bring this conference to where it is. So, and I hope, we could keep celebrating these anniversaries over and over again. There are many opportunities, and just where the field is going, it's just, it's a pleasure to see conferences like this being, vibrant and just the tenure that they've had. So, what I wanted to do, to clearly, just to clarify, I'm not a mass spectrometrist by any means. It's just, I got into mass spectrometry purely out of interest and just sort of seeing the power of the technique. But the number of you here attending know more of this than I ever will or ever could. So, a lot of my presentation, really looking at the topic, you would wonder why you'd want something like this at a mass spec meeting. But what I wanted to talk about really is where sort of the whole manufacturing technology is going, where processes going, and so on. And really, I think through the talk, you would be the wise ones to sort of figure out where are places where mass spec can come in. And there are a number of them. I'll show you a couple, but this was just more so for generating some of that discussion, because I feel, with all the sophistication in manufacturing, the analytical is picking up too, but I firmly believe that mass spec is truly where things are going to be moving forward. So I'll start, I just want to reminisce a bit of the past. This was the very first meeting we had, and just wanted to show you, this was, as I told you, something we thought, let's just get together. CAS was really kind enough when I had put a proposal to say, let's start a meeting like this. CAS right away sort of saw what the value was, and they were really generous enough to sort of say, let's give this a shot, see where this takes us. And number of people over the years, there's good friends here that I've had the pleasure and the fortune of working with in different roles. Many of them are very different roles right now, but this is what the meeting looked like. And as we were putting this together, interestingly, I had shared this when I spoke in 2013. A few days before the brochure was supposed to go out, I got a call from Karen Bertani, who's was organizing things for that meeting, and said, we need a logo So what are you talking about? So no, we need a logo. Every meeting has had one. So at that time, I was working in the lab. So I said, okay, let me just go in, and this is what came up, like it or not, but this is the best I could do in the 10, 15 minutes I had, and see that it still continues. So that's a little bit of a history in terms of where all this started and where it's come about. So, again, it truly is a pleasure speaking with you guys, and I really thank the organizers for this invitation. Let me start by saying a few things about where our company is, Biogen. I think, as a number of you know, we over the years have focused almost all our efforts right now on neurology, and in various areas from things like movement disorders, certain aspects of pain management, dementia, and so on. And what sort of became evident very quickly is, in order to be in this space, given the disease areas, just devastating disease, the number of folks that have been impacted by it, the impact that it has had not just on the patients, but the caregivers, the families, and so on. These are large indications requiring reasonably high doses. And what became very, very evident at that point is we had to evolve our production platform, not just how we developed it, how we manufactured it, because there was only certain limitations. The idea was we couldn't just keep on building large-scale facilities. We had to come up with very high productivity processes that could be monitored well, so that we had a reasonable rate of success, and almost an end-to-end change in terms of how we were operating. So a lot of this work began must have been 7, 8 years ago at this point, maybe even longer, where we started revamping the processes to where the platform is right now. But before I go into that, just to give you a sense of what these numbers look like, I mean, it really is, it's a devastating situation here, with causes of disability, what the neurological conditions do, the number of patients that are impacted by it, in some cases, the life expectancy associated, and just sort of, the fact that there aren't as many promising therapies, and a number of them coming up, including from our company and others. But this is something that sort of is what's driving all of us to come up with all these new techniques, technology, and figuring out how one can really be able to fulfill this unmet need. So I just want to give you a little bit of a premise of this, and much as what I'm going to talk about is there's a need that has driven this. This was not just technology being developed because things were available, right? So our network, I mean, I'll get into my talk right now. Just given the indications that we have, given the risk around some of these things, these are not easy to kind of address. So we, right from the beginning, this has happened for a while, we really went into a very flexible model for operation. So we have sort of, fortunately, what we call a flexible factory, which is single use at 1,000 liters. We have stainless steel after that at 2K and 15K. And clearly, all this is evolving. There's a lot of interest in continuous as well. And I do believe that that's another way to start optimizing your network. But at least from a flexible, number of you folks are aware, and a large number of our peer companies are in this situation as well. It does offer a lot of flexibility, and that's how we're approaching it. The way we're developing our processes right now is right from the get-go, we develop a commercially viable cell line. So in almost all cases, I'm talking for biologics, and a good amount of our portfolio are antibodies, is that we develop a high productivity process. So right from the get-go, it will be 10 to 12 grams per liter. As it gets into early phase manufacturing, phase 1. The idea being that the technology is such, we have screening technology and so on from the cell lines, that this is not something that's coming in the way of starting tox studies and so on. I mean, many cases we may not have the single clone and so on, but this is how we approach it. We go straight from production or straight from development into a single-use system, and then in most cases it'll go up to large-scale production. In most cases it'll be scale up from 1,000 to 15,000 liters, and that's sort of how we've been operating for many of our products. So this just gives you a little bit of a snapshot of sort of where our production is, and clearly the pros and cons of single-use versus stainless steel. I think this has been talked about for a number of years. There's been a lot of discussion, in conferences. What is the right, what's the optimal model? And frankly, my position here is it really depends on what you have as an infrastructure, what is your portfolio, and that's how we've been operating as well. So, and clearly, when you look through all these productions, all of it eventually comes down to cost of goods based on really optimization of your manufacturing facilities. And these graphs are in some ways self-explanatory. As the throughput goes up, optimization of the overall productivity of the facility, and the results in cost of goods impact as well. So what I'm going to talk about, a lot of what we are calling next generation started with this facility that went into operation last year. And this is a facility located in Switzerland. Much of the work that we've been doing in my organization. So, I mean, if I, as was indicated, my organization is responsible for developing all the products in our entire portfolio, biologics being one of them. But when we started this work, the idea was that this would actually, we targeted based on this facility coming up. So in fact, it gave us sort of a bit of a playing field here to figure out how do we pull all these things together. But the facility is really set up in terms of cells. So you have 2 seed trains feeding 4 18,000-liter reactors, which would then go into one single purification train. So each cell is what I've indicated here. There are about 2 cells right now, and they're common facilities like warehouses and so on, feeding into this. But the idea really is coming to each cell based on where our processes are, 10 plus metric tons coming up. And depending on the way the portfolio moves and the needs come up, you can start building each cell. In most cases, you could duplicate it, or you could have a very different design. And it doesn't all have to be necessarily proteins. It can be gene therapy or what have you. So it really gives us a lot of flexibility. And what we've done is implemented much of the automation, a lot in terms of how we're running our processes, which I'll get into a minute. All of this incorporated into this facility. And this in some ways is becoming the standard. So our other facilities are being modified accordingly, according to where we are with this. So this just gives you just a brief snapshot of what I'm going to talk about, and a lot of the tools that I'm going to talk about are related to this. So I had shared this slide in 2013. This is really where, at least my assessment was, right or wrong, where things were and what the future expectations would be. I won't go through this, but there was a lot in terms of better understanding the levers that affect product quality, so that you kind of build product quality, this term that has been used so much over the years, that truly build product quality. And there was a lot of, as you guys may be, and a number of folks have been involved in this through the industry, this is not just us, a lot of use of mass spec and so on, a lot of online monitoring when we're doing some of this development work, and even in some of the manufacturing areas. We've also sort of had said, we would need to invest a lot in terms of raw materials, because around that time, chemically defined media and things were kind of coming up. And it was very clear that subtle changes did impact overall performance of the process. So that's something, and companies have invested a lot in that area, better understanding of process consistency beyond CQAs. And one area that I wanted to talk about, because we had started work when I had presented this, was on predictive modeling. And this is not just sort of predicting small scale performance to large scale, but what I want to talk about predictive modeling today is is generating a lot of data through development and manufacturing, and then being able to have predictive signatures that will actually be able to predict product performance. Product performance based on outputs of your process. Some of it may involve actual measurement with tools, could be simple tools like sensors and so on, could be far more sophisticated tools, getting into the area of mass spec, even 2D chromatography and so on, but then eventually using models. So this is what I had said we were working on, and this is where we are fully embedded in right now. I won't talk much about the product and so on, but nonetheless, having talked about this is really where I feel things are. And a number of companies over the years have shared where they are. So many of our peer companies, I feel, are in this space. This is nothing necessarily unique to Biogen. But we do have a number of tools, I'll chat about that in a minute, where raw material screening has just become it's been such sort of a beneficial exercise when we approached it the way we have. We truly have a good understanding of what the control mechanisms are, whether that be with raw materials, whether that be with certain process levers, how they eventually control product quality. The linkage of unit operations, we started on with QBD, which was the term used many years ago, and a lot of the linkages came from that. But an area where we had talked about, I would say 7-8 years ago, was our desire to do feedback and feed forward adaptive control. This was real time, done in as automated a manner as we could. And this is really the platform that we're evolving for many of our biologics. And I'll chat about that in a minute too. And this has been implemented right now in our facilities, and which will be for commercial production as well. So, clearly clinical, but then commercial production too. And for that is extensive real-time monitoring, some of it by measurement, some of it through models and so on. And sophisticated models to a point where we have been using models for biologics for some time now. And we had actually filed for using these models to predict quality in place of actual testing. So this was a successful endeavor in terms of using that. And this is sort of where I feel things are. So I think what's nice is, as an industry, we had talked about this. And what's so encouraging is that we have somewhat kept up to our promises, and we are at the stage where we can say, a lot of these things we had talked about a number of years ago are being implemented and working quite well. So having said that, our general approach now is the way we are approaching all our processes, all our production, is that we're trying to develop as clear a connection as we possibly can to process parameters that affect quality. And then, as I said, models come into place, process signatures, looking at growth curves, peak shapes, and so on. All of this eventually to sort of say that we need to have full control. In other words, what we're saying is, for all our unit operation, this is something I strongly believe in, and which is something, as unfortunately the company we're proceeding is, we want to understand all the way from looking at our production reactor, all the way through bottling, what is actually happening to the product, and this has nothing to do with saying based on this we'll eliminate testing or release testing or all. It's not so much about that. It's purely getting a sense of how what's happening to the process, so we're not looking at it at the end. We're kind of analyzing it from where it's necessary, where processes, certain unit operations impact the product quality, and for us to know what's going on. And I said, starting from the reactor, looking at it over the time frame in a reactor, not at the beginning and the end necessarily, but really doing a time course, figuring out things like how is glycation changing and so on. So that's really where what this eventually comes down to. And some may ask, why would you want to do that? And this goes back to our production, right? If we are producing at that high a volume, we are seeding reactors very frequently. So what we're trying to get to here is be in a situation where there are no surprises. So release testing just confirms what we already know. And the idea being that by knowing where the product is going, I mean, there may be certain unforeseen circumstances, we want to be able to curtail a batch, because not doing that, many times with the rate of production, you can very easily see 6 or 7 batches, not working or, having failures if we don't intercept this. So that's really where all this is leading us, and I'll talk a little bit more about that. So this is just want to give you an overall picture, starting from raw material screening all the way going through upstream and downstream. This and filling, in this case, showing you with bags. This is what it looks like. With the raw materials, as I've said, we started working with vendors and trying to sort of establish this is what we would need from them, and then establish some of these techniques at the vendors. And then really when raw materials comes to our door is looking at some very rapid tools like handheld Raman analyzers. And at this point, pretty much everything that comes in has been going through some kind of rapid screening. And eventually it really comes down to, it's not just that, where then some of the modeling comes in and so on. A lot of the data analytics come in is really making that connection from raw materials to manufacturing outcomes. And this has been, I can't say enough, this has been extremely, extremely beneficial for us, be that through normal production issues, any investigations that come up, having this kind of tracking has helped us. And as I said, with bioreactor feed control is the same thing, that our idea is that fine, you could, many of our processes early on had been bolus feeds and so on, where we have sort of fixed timing. And we're really moving all that through, and I know other companies are doing similar things as well, moving to based on actually measuring and figuring out when the feeds be. So it's almost like an adaptive thing. And the idea here is really based on this, getting a consistent product output, performance and quality output. And it's not just to the upstream, but even as you come to downstream, it's looking at sort of feed forward controls, figuring out how one can control the outcome of downstream processing. And that's sort of something I'll chat about a little bit too. It's in this particular case, I'll show you an example in this talk as to how we control aggregation, based on measuring before the load and then figuring where we take it there. And then looking at, inline refractive index control of the UF, protein concentration measured with all kinds of tools, solar VPE and so on. So it's really, this is giving you a snapshot of end-to-end where things stand. And clearly there are a number of other techniques. I'll show that as well in terms of bioreactors. What tools are we using for looking at performance of reactors, being able to monitor growth rates and so on. So this is just a overall snapshot of what is in the new facility coming up and how the other facilities are adapting as well. Right. This is maybe a bit of a better schematic, looking at it where the feedback control and feed forward, we're looking at it all through raw materials, upstream and downstream. So even in the raw materials, figuring out, based on our understanding of what the impact is, in some cases may need to fortify them, so sort of get to that consistency. But the bioreactor, I'll show you an example of how we control glucose as one case, and can be a number of other applications. And as I mentioned in the downstream, looking at, product attributes, I'll speak in particular for aggregation in this case. But this is really how we envision a process. This is how we've been presenting this to regulators as well. And all of this has to be, eventually comes down to really understanding what the inputs are, what the impact of the output is. How do we really get the processes to a point where we get them as narrow in terms of the consistent output? And this is, it's been working well so far. Yeah. So here is an example for the downstream, just sort of talking about basic tools. The tools in this case are very basic. whether you have capacitance probe, in this case, is talking about Raman probes. And, that's a decades-old technology. This is nothing necessarily novel, earth-shattering here. But what we have found is with basic tools like Raman, there's a lot of information. This is, even times when I was in the lab and some of these things were evident, relatively low background and ability to monitor a number of things, and we're talking about glucose here, lactate, what have you, But this was the advantage of using sensors like this, is where in a complex spectrum, you are simply looking at areas where some of the differences could be. You can do a lot of this work by simple training experiments. But once you find these differences, then it's a matter of using all the other sophisticated tools that are used in manufacturing, conversion of the data and so on. And the measurements are actually extremely accurate. What you see in the box below here is just looking at how the measured and the predictive models are. So this was actually measured, and based on some of this, looking at some of these signatures, being able to predict what the outcomes are, to predict what the outcomes are, and then to feed the reactors as and when glucose is needed. So you're truly, to a large extent, moving away from bolus feeds. And what we found is a much, much better control of the output of a process. So as I said, a lot of it at our new facility is no manual intervention at all. In some ways, it's as flawless as it goes from a pure adaptive feedback control. And speaking a little bit about the modeling, a lot of the modeling here starts from very early stage development. I mean, whether you're looking at a CFD type of modeling, we are looking in terms of in silico, sort of figuring out how we develop processes as well. Process modeling around looking at inputs and outputs. But an area which I'm particularly interested in, and we've started to do quite a bit of work, is from a facility. We talk about all these things about controlling outcomes of processes, but the outcomes are controlled based on the instruments you use. And it could be very simple in terms of a pump, pump that's used for pumping, critical materials, feed streams, and so on. And even looking at pumps, even looking at certain valves and so on, I would like to kind of shift this. I've presented this at a couple of meetings already with some of the work that we've done, is even using things like pumping speeds, looking at pump performance, looking at the vibration from a pump, and being able to predict what the performance would be, what could be potential failures, how do we predict that in advance so that you don't get all these expensive costs due to breakdown and so on. And some of it may come to even things like valves and so on, being able to monitor flow and figuring out where some of these things could be failing. So modeling here is in a way, what I wanted to show was an all-encompassing thing. It was not simply saying, let's look at what the product is, but it's looking at the facility, layout of the facility, operation of the facility, figuring out how do we make buffers, our concentrates based on what our process is going to be. Is that an optimal way of doing it? The entire operation mode on a facility. And so at least in my organization, we have a dedicated analytics group that's looking at this end to end and trying to figure out how we can utilize this. It's fascinating, as sort of, although there's so much work to be done, but just in terms of data that's come out so far, I think it's so beneficial. And this really goes down to the whole area of digital that we are talking about across our industry. So this is just, I won't spend a whole lot of time on it, but this has just been our legacy processes, where we had, certain variable inputs and so on. And in some cases, I'm not saying that output was totally variable, but not quite controlled to the way it was, where we had real-time monitoring and really an adjustment of a process, which is what we truly are defining as an APC process, with the idea that you can get a very consistent product quality. So in simple terms is, if we control how the processes are moving, whether that be in terms of growth curves and so on, you would get consistency in terms of product titers, how the cells were performing, and eventually product quality. So this is really how we are approaching this whole idea of advanced sort of process control. But when you start looking at sort of some of the principles here, I mean, just want to summarize what I had shown before. I talked about the raw materials. I won't spend more time on that, the actual process control, but the process product testing, which may be relevant, really, I think it's for this meeting, is, as I said, we talked about sensors and more sophisticated tools. Our idea is not so much in terms of this whole real-time product release, where you eliminate release testing and so on. The idea was really, so when we implemented testing, was purely to get a better sense of product and process performance, product output and process performance. That's what we're trying to do here. So and testing at the right points, testing at a points where in fact you can impact what the outcomes could be, rather than testing later on when after the fact there's not much you can do. Either you have a batch that's not quite in performance, or you may have to reject that. So that's really what it comes down to. I just want to kind of frame that properly. So it's not so much about necessarily eliminating release testing. What I said earlier was, when we do testing at the end of a product, it is just to confirm what we already know. And that's sort of in the principle that we're operating under. Just want to quickly show you sort of just the model evolution of models, and a number of you who are far more well-versed in this than I am would recognize and appreciate this, but it takes time, and this is something, for folks that may be looking at implementing this, whether you generate some, incredible mass spec data and using that to model things, it is a journey, and sometimes the models aren't perfect, and one has to be realistic about it. You may have to cut your losses and move into a certain different approach, but to a large extent, what we have seen is the models have actually served us well. And I just wanted to kind of I don't want to go into the details here, but just outline just the journey along, starting from a very early stage where we were building in, training sets to create models. There was a good amount of testing required, as experimental design and so on, to then starting to use some of the tools, the PAT tools, the process analyzers. I showed you some aspects of that. And then getting later on, as far as now looking at pattern recognition, where the model truly starts developing, where you start seeing differences in patterns, and then being able to take those differences and implement that towards figuring out what the product output would be. Interestingly, this pattern recognition, we've been working on it for a while. In fact, I had been talking about this in a couple of mass spec meetings, where we're looking at complex spectra and so on, being able to conduct pattern recognition on that, and being able to kind of start looking at what the impact on product quality would be. So this has been going on, I would say, probably at least for the last 15 years or so. And then eventually, as I said, the last stage is the predictive control, where the hesitation in some cases of looking at a lot of online analysis has been, how long do you keep doing that? And the idea is that this is not something where you just continue on. It's just you start the testing and you then gradually move to other modes of predictive control, that it doesn't all have to be around testing. And that's sort of where we are as well. So this just gives you an example. I actually want to show you, some of the data that we've been generating. This is from not too long ago, but this was looking at, how do we monitor glycation in this particular case? And this is making sure that you look at the glucose at a certain maintaining low levels of glucose, which has been linked to its impact on glycation, or a high level of glucose where you step it down over a period of time. But this is, again, using the right kind of sensors, in this case, it was Raman, and using those sensors and being able to then make sure we feed the reactors in order to get a controlled experiment. This was essentially a lot of what I'm showing here was from some early experimental work to figure out how one can actually control some of these critical inputs into a process. And this goes with almost any other inputs that you would have through the feeds and so on. It's, again, as I said, basic tools that are providing a lot of impact. In this case, it's glucose and lactate control. Yeah. And what I had said earlier, what you see in the blue box above here is something that the reason we're doing all of this is consistent cell growth, which has then led to consistent titer and product quality, And it's been quite impressive with the data we've seen so far. I want to just touch a little bit upon downstream. Downstream, again, I've seen a number of talks over the years, very, very good data. Folks that have talked about Amgen has talked about a lot of the multi-attribute method control, and I think it's really quite powerful. But a lot of our work, I talked earlier, was online sampling, aseptic sampling, but then downstream has an important impact, and some of the upstream clearly is impacting the downstream, and I'll just show you that in a minute as well. But same thing with the downstream, trying to be able, our idea is column to column, look at what the column is impacting, and can you measure it, can you predict it, and as a result of that, can you then intervene and bring the process in control, And I just want to qualify this because, when we're talking to regulators also, when I said to bring a process control, doesn't mean that the process is out of control. We're trying to tighten it even further. That's what I'm getting at. They're basic tools to do that. And plus we're getting a sense of where the product is and where the process is, and the process is all through. So that's really what this comes down to. Let me just go in. This is an example I had said earlier in terms of a feed-forward control, looking at a column that helps us get to the aggregate levels that we want in this case, or it could be any other impurity. And based on what you come up with the output of the previous column before it's loaded on to the next column, in this case, polishing column 1, you could do a number of things. In this particular case, it's changing the loads, based on what the impurity levels are, you can then change the loads, which is what we would do in our normal operation, change the loads in such a way that the impurity levels would be at where we want, and this has really enabled us to have tight controls. In some cases, the aggregate controls levels or certain impurities have been much, much lower than what these numbers are. But this is exactly another point talking about truly affecting or impacting outcomes. This is, again, I won't spend so much time on this. This is just giving you a generic picture of what the outputs are on the production floor. In this particular case, you have your operating limits, what we have seen historically, how a certain product quality changes over a course of production. And this is then looking at what some of the predictive modeling shows us, what the actual modeling shows us. The correlations are really very good. But this was a model that has evolved. This is not what came up, in 2 months or 3 months of production. There was a lot of online monitoring, and based on that, some of the models evolved, and the correlations, as I said, was good. And as you say, things that I've been talking about, the release testing really confirms what we already know. And this is, not to get into CFAs, but this is really what we're trying to get to, is monitor things where, in fact, they need to be monitored, where it truly is impactful, eliminate this whole thing of waiting all the way to the end, and then get into this area of, using the right tools to confirm what you already know. In this case, it's, looking at product quality, whether this be through multi-attribute methods, looking at binding from the bioassay and bioburden and so on. So this is really where things go. A lot of this has to do in terms of managing how you control your disposition, because going through very traditional ways can be a long time before QC labs go through the whole thing, versus the way we're approaching it now, really cutting down the time around production, and when we truly can say, okay, the batch is in very good shape here. So this was, again, just giving you a snapshot of some of the things, where mass spec has come in, particularly as we kind of moving to other areas, maybe continuous manufacturing and so on, where in fact, I feel this is even more critical. Using the right kind of sampling through a number of sterile ports you can get, having sample processing there if you need reducing conditions or not. But then the way we've approached it is looking at all the sophisticated techniques we have to understand what's happening in the reactor. And I had said earlier, this could be 2DLC, looking at glycan analysis, looking at intact mass, reduced, and so on. And this is just what we believe eventually PAT, benefits and how that helps in our operation. Okay. So some of the last couple of slides. So the traditional approach, things that we've all been used to, what we're trying to do through this is not so much sort of saying, PPQ is important or not, but the approach that we're really trying to get to here is that the right kind of process design and understanding eventually gets you to, what we would like is a continuous verification. How much goes into that will change over a period of time. But our idea is eventually, so much not get so hung up on the PPQ aspects and say that, okay, you've gone through a PPQ, it's great, but really this idea that there are tools available, there's knowledge, historic knowledge of hundreds and hundreds of antibodies that have been produced in the industry, so many produced from our company, where we need to get into this idea of from a process design, then as you go into production, it's truly continuous verification, and it's giving us a better sense of what the product looks like. And this is really where I said, you guys can truly make an impact with all the knowledge that you have, all the advances that are happening in mass spec, the ability to detect things, intact levels, the rich information that's coming in, the high resolution systems that are coming in, quick inputs, outputs, and the fact that mass specs at a point where, you don't need, somebody so highly trained. There's ability to train a number of individuals in this, just the way the technology has evolved can be a tremendous benefit here. And our new ways of working are precisely this. We are going into, just based on our portfolio, very, very high productivity processes. The throughput to the facility is what we focus on, as I mentioned, through modeling and so on, putting in the right kind of controls in there, and not being so concerned about putting these tools on the floor. It's nothing. It's a detector to a large part, whether that be a mass spec, whether that be a UV refractive index. And if it gives you the right information, we're willing to do that. And just truly high performance operation, localized testing where it's needed. You're not shipping samples all over the place, or you're not doing a production and then eventually testing 2 weeks later. All of this is done in a way to optimize the operation. And through all of that is clearly, systems need to fall in place. So we've gone into a very recipe-driven operation, facility to facility, having it as consistent as we possibly can, although there may be some differences in equipment, depending on how the facilities came about and so on. Decisions being made on the floor, a lot of sort of setups in our, particularly in new facilities, is where you have proximity to labs and so on. So everything doesn't necessarily need to sit on the floor, but the accessibility is there. So a lot of quick interactions, personnel thinking differently, and just getting to a different phase. And, to support all of this and what the impacts are, in terms of reduction, in terms of issues coming up with batches, to a reduction in batch review timings, process robustness, testing, and really optimized facilities. So it's the impact is clearly there, as far as how we operate, how the investigations are run, because if you have data at hand and accessibility, a lot things can be very different. So here is the future, and I think it's not even so much the future as in what the present is, which is going to evolve. Much greater understanding of process, understanding control, predictability coming in. I truly believe this. I would like us to be at a stage where manufacturing is like a control room. I mean, you don't have folks sort of all over the place, but through a control room, looking at the signatures, being able to predict outcomes and control it truly in an adaptive and automated way as it can be. So this is happening now, and I think it's going to evolve. This is our facility where all these things have happened in Switzerland. And lastly, I would like to acknowledge, none of this is my work. I wish I were that brilliant, but there are a number of individuals, departments that have been involved. And in closing, again, thank you so much to the organizers and all of you that have been so influential in keeping this meeting going and, and with all this energy. So much appreciated. Thank you all.
Thank you, Rohin.
Thank you. Excellent talk. So we have a question. Since multi-attribute mass spec methods can generate rich data on product quality attributes, have you been able to incorporate MS data into the multivariate predictive models of process performance?
Yes, I mean, at this point, it's any data that actually helps us. We would want to do that, A lot of it started initially with, using things like 2DLC and so on, but increasingly, as you saw, use of mass spec and the multi-attribute methods, absolutely. I mean, really the models, the key, I mean, there's no rocket science, is having the right kind of data and really rich data to be able to start building these models. Because many times the models, I mean, they're random associations that can help come up with these models too, So, yeah.
Here is an interesting question. Do you see an application of blockchain technology currently mainly applied to cryptocurrencies to the biopharma production processes?
So you broke up initially. I didn't get the first part. Is there?
The blockchain technology, do you see the application in the biopharmaceutical production processes? Sorry.
Yeah, I hadn't quite thought of it, but yeah, why not? I mean, at this point, what's so interesting, from way things have evolved is I actually don't doubt anything, to be honest, because the possibilities are so endless. I think the approach that I'm asking my organization is, in a way, be as open-minded as we possibly can. You never know when these connections would come up and where that could lead us. It's an interesting question. I don't have necessarily a good answer, but I would say, worth thinking about.
So I have a question, Rohin. The application of metabolomics to cell culture, do you see that there's some utility in understanding metabolomics and potential biomarkers to cell culture productivity and cell health, and being able to use these as readouts to extend cell culture in the future at the process scale?
Yeah, I would say omics in general, to be very, very heavily invested in that. I mean, I'm not as close to it as I was a number of years ago. So I wish I could give you a more concrete answer. But if you may not recall, but in 2013, when I had spoken at this meeting, one of the things I'd emphasized was some of the work that was going on in omics. And at that point, it was primarily proteomics, but beyond that, we have expanded to even lipidomics and so on. And, I've had people in the group work on their pieces for certain applications that we wanted. So I would say yes to that. I can't sort of quite tell you specifically what may be going on right now.
Thank you.
Great. Well, we are waiting for the other questions to come up. I have a question about the multivariate analysis work you have done. It looks very impressive software prediction. My question is, do you guys drill down to those measurement attributes that cause the process variation and then look into the root cause of that, maybe from the biology point of view?
Yeah. So I think we start, the two aspects to it. One is the development history has given us, reasonable amount, because to a large part, there are certain platforms that we're using. And, we really, when we look at platforms, we take it seriously, not to say that all processes are identical, but there is a lot of process knowledge that transfers. So we look at it right from the development stages, but then go back based on the manufacturing history to make those connections too. So absolutely, I think really key at it is really getting to the root cause and then figure out how do you then impact. One is the root cause, but the other thing which I really feel where has been very helpful, and it's been a bit of a journey, is to figure out, are there ways in which you can self-correct? It may not be the case always.
Yeah, thank you.
Another question from the audience. Can you give the snapshot of continuous processing and where do you see it going next?
Yeah, so I've always been very fascinated with it. And it's not just even for proteins, even for certain small molecules, because they're far ahead in terms of continuous drug product production and so on. And not to say that, we are not, at least. I actually do see a place for it. And, with all the tools coming up with, things like better designs on ATFs and so on, I do see a place for it. What's interesting is I feel, from your own network strategy for an organization, it's a very, very good tool to avoid large scale facilities being put up. There are limitations to it, of course, because one has to remember, because when you start looking at cost of goods, when you come to about a certain sort of production output, continuous may not be as attractive as a stainless steel large scale production. So the way we are approaching it is to sort of say we need a toolkit for production, and continuous will be one part of that. And there are certain applications when you look at the output and the cost of goods. And the fact that you can put up these facilities, when you compare them to putting up large-scale facilities, the cost, the timing, I do feel that this is something that you'll see more and more happening. I mean, the number of companies that are already doing this, even up to a 2,000 liter scale.
Thank you.
Yeah.
Great. Yeah, I just want to remind everyone, please type your question in the Q&A box. While we are waiting, I have another question. So I noticed you have been very cautious on real-time release testing and use right-time release testing. And the future CFA looks very encouraging and promising. So the question is, in your mind, how far away as an industry from that automatic generate CFA? How many years before we can get there?
Yeah, no, it's a good question. I think at least we are approaching it, and we've spoken at various meetings and so on. When this whole real-time release thing started off, there was a lot of desire to kind of remove end release testing. Yeah. And I mean, in reality, you can do that, but I think as an industry, we need to maybe take a different approach to it. And that's what I was trying to allude to here. Is real time, I mean, testing at some point, shipping all over the world and all that, some aspect of that, is necessary. But we need to get a sense of, how we controlling our product through the whole production cycle. So as I said, risk of repeating myself, so that the end testing simply confirms what you know. So we are not going down the path of saying, let's eliminate all this testing and so on. We're testing in different places. And, in some cases, as I said, we're using process signatures and models that come up in place of testing. And that's how we're approaching it.
Great, thank you.
So I had another question, Rohin. So the very first conference, I know that it was first mentioned whether or not mass spectrometry had a place in the quality control laboratories on the subject of testing. Again, in 2013, on the 10-year anniversary, there was an entire session that was dedicated towards the possibility of moving mass spectrometry into the quality control laboratories. Here we are 8 years later, and I wondered what your thoughts are on still that prospect of mass spectrometry as routine analysis in the quality control laboratories. And I do believe that it certainly has been implemented at some companies. Wanted to get your thoughts on that.
Yeah, I mean, this has been a bit of a humorous discussion over the years, because it's sort of kept coming up so many times. And I feel in some ways, I'm not trying to be facetious here, but if we had actually implemented it all the time that we spent talking about it, we would have been so further ahead. But we bring up a good point. In all seriousness, I really feel, I don't know what the debate was. There may have been some anxiety about using mass spec, cost, maintenance, what have you. I look at it as a critical piece of understanding anything about product. We use it. I know several other companies do. And it's become, a detector with a lot of data generating power to it. And I frankly think, if we have to progress as an industry, not to say that we haven't, we have to embrace this. We have to embrace things that actually help us get a better sense of the process. I know there are sort of different views, and I'm speaking more broadly, where we feel that, why do we need to put in all these tools? You can just develop better processes, make it as simple as possible. And I stand in a very different camp. I said, there's only so much you can understand of a process. And this idea that you will just keep developing robust processes in some ways is maybe a bit of a naive assumption, is that we need to have the right kind of tools to just help us better understand the process, because there can be differences. Process variability comes up, years after a process has been commercialized, where you truly start seeing that. So a long answer to it is I really feel that this instrument is there. I don't think there's any pushback from regulators, at least what I've heard. And it's a powerful tool. Why wouldn't we use it?
Thank you.
Yeah, I think you more or less answered one of the questions from the audience regarding to what's the limiting factor for MS taking a more prominent role in the manufacturing space. Yeah. So let's go to the next question.
All I would say to that is unfounded anxiety.
Yeah. So next question is about modeling, right? So can you please comment on acceptability regarding to the use of predictive models in GMP environments? For example, classification readout generated from a black box, quote unquote, algorithm process. How well do regulatory agencies accept them?
So far we've seen there's a good acceptance. I think it's not necessarily an easy discussion as you get into it, because to a large part it's a different concept. But I think our approach for modeling in the GMP is working because we're using modeling to predict, to control processes, and it's not so much to drop test and not to do release testing, although we had filed on this and it was successful. The large part, I think modeling needs to be used for what it actually provides. And it's moving beyond process, as I said, when we're using it for modeling, equipment, facility design, and all those things, that's where I see use. But, done right, if you can show the correlations, not all models are going to work. Some correlations may be weaker than others, but we have seen a number, and, in our filings, it's a matter of you may get questions, but the data will eventually speak for itself. I think that's what it comes down to.
Great, thanks.
So the next question is, where or what do you see are the biggest challenges to implementing APC?
I think, beyond the technology, the biggest piece I feel, and we're having discussions recently in terms of this whole digital evolution as well, is the cultural aspect in an organization. First of all, most important thing is management needs to kind of see this and being able to stand for it. It takes a certain level of courage. You're taking on certain risks. But following that, I think it eventually is a cultural change in the organization, because a number of folks that have worked in this field, very knowledgeable and talented, are used to a certain way of working. We had to go through that. There's a lot of manual interventions, and with that there was success rate, but it was not an efficient operation. And that's sort of where things like APC, other types of digital tools that help. Eventually it goes down to, and this is not such a hard business case to develop, it will lead to a much better throughput of your facility, in terms of success rates, in terms of how you actually operate, and knowledge that comes out of it. So, yeah.
Thank you.
Great. So the next question from the audience, have you connected your process modeling efforts back to the sequence variants at the candidate selection stage to predict where your best process levers might be?
Yeah, I mean, that's a really good question. Not quite, I would say, as systematically as one could, and it may eventually get to that. But right now, the initial screening, a lot of it is, when we're looking at sequences, much of it has to be around expression and stability, in terms of the formulation and so on. But yeah, I mean, that could be eventually, as these things mature, it's the possibilities are endless, frankly.
Great, thanks.
Our next question is, good predictive models usually need a broad range of data for training, including bad process situations.
Yeah.
Controlling the processes to be mostly good, how do you generate the bad situations to improve models?
Yeah, that's a very, very good question, actually. That's why I sort of alluded to earlier. Some cases, it's really using the right kind of training sets to generate this data. Some cases, actually monitoring, putting this monitoring, which is the evolution of the model itself, using monitoring tools all the way, early stage before a model even develops. And some of the process variability and all those things tie into that. So it's, that in combination to some of the machine learning, which was one of the slides too. So it's a very good question. I don't know if there's necessarily a generic answer to it, but it depends on what you're trying to model. In some cases, you could create some good training sets to start building these models. In other cases, it may have to evolve based on more data that you gather.
Thank you.
Great. I think that's all the questions we have. Chris, did you see other questions? Or?
I think that's all the questions.
All right. With that, I think we're reaching the end of the time frame as well. I just want to thank Rohin for a very visionary talk. Thank you so much for taking time and come back.
Thank you.
Very good, delivered an excellent keynote presentation.
Yeah, and keep pushing for this technology. Believe me, it is the most powerful tool we have in our hands right now. Yeah. And with all the progress made, I think I'm just looking forward to what's to happen in the next 5 to 10 years.
Thank you very much. Yeah. Thank you. And also I want to give a quick reminder, please take your time to visit the posters as well as vendor booth. They're virtual. I have been there. It was a very good experience. Please take time and appreciate our vendors for the support as well. Thank you so much. And we are moving to the technical seminar for the next session.